Bad RNA quality can ruin your entire sequencing experiment. So how do we prevent that? Let's compare the essential QC tools you need to know. Qubit is the tool for accurate nucleic acid concentration. Qubit uses fluorescent dyes that specifically bind to DNA or RNA, which means it ignores contaminants like proteins. This gives you a much more precise measurement, especially for low-concentration samples. That's why Qubit is essential before library prep. It ensures your input amount is correct and improves the quality of your sequencing results. NanoDrop measures absorbance to estimate nucleic acid concentration and, more importantly, assess sample purity. Key ratios like 260/280 tell you if proteins are contaminating your sample, while 260/230 indicates other contaminants like salts. It's fast and requires only a tiny volume, making it perfect for an initial quality check before moving to more accurate quantification. The Bioanalyzer is used to assess RNA integrity. It separates RNA fragments and generates an electropherogram, showing peaks like 18S and 28S ribosomal RNA. From this, we get the RIN score, which tells us how intact the RNA is. High-quality RNA shows sharp peaks and a high RIN, while degraded RNA appears smeared. This step is critical because poor RNA integrity can severely affect your sequencing results. qPCR is used for library quality and accurate quantification. Unlike general concentration methods, qPCR measures only amplifiable DNA, which is what actually matters for sequencing. It also helps detect issues like adapter dimers or poor library complexity. By giving you a precise estimate of usable library molecules, qPCR ensures optimal loading onto the sequencer and improves overall data quality. Next, we check fragment size and library distribution. This tells us whether our DNA fragments are in the correct size range for sequencing. Ideally, we want a tight, consistent peak, showing uniform fragment sizes. If the distribution is too broad or has unexpected peaks, it may indicate issues like overfragmentation or adapter dimers. Getting this right is crucial for efficient sequencing and reliable results. We can check for DNA contamination in RNA samples using methods like agarose gel or control assays. This is important because leftover genomic DNA can interfere with downstream analysis and lead to inaccurate results. Unlike traditional PCR, dPCR partitions a sample into thousands of tiny reactions, allowing absolute quantification of DNA without a standard curve. By counting positive versus negative partitions, it provides highly precise and sensitive measurements. This makes dPCR especially useful for low-abundance targets, rare mutations, and validating sequencing libraries with high accuracy. Finally, sequencing-based QC uses a pilot run or shallow sequencing to evaluate library quality before full-scale sequencing. By generating a small amount of data, we can assess metrics like read quality, mapping rate, duplication levels, and bias. This helps identify potential issues early, saving time and cost. It's a powerful final check to ensure your library is truly ready for high-quality sequencing. This table summarizes different quality control methods used in RNA-seq library preparation, along with their strengths. Each method serves a specific purpose, and no single tool is enough on its own. For example, NanoDrop is great for a quick purity check, while Qubit provides accurate concentration measurements. Bioanalyzer helps us evaluate RNA integrity and fragment size distribution. qPCR and dPCR offer precise library quantification, especially for sequencing-ready molecules. Finally, sequencing-based QC, like pilot runs, gives us the most realistic assessment by testing actual sequencing performance. The stars here represent how strong or reliable each method is for a given purpose. Overall, combining multiple QC methods ensures the highest data quality and more reliable sequencing results.